And now to your question about the exclusion zone. It's a unique test bed or test field for radiological and radioecological research. Even after the Fukushima accident, the contamination was with cesium, not with strontium-90, plutonium, americium, etc. So we had very high levels of radioactive contamination. The maximum values of the dose in the so-called red forest are about 500 microgray per hour. In Fukushima, it's about 8 to 10 micrograde per hour. So the Chernobyl zone is unique in terms of obtaining new knowledge that cannot be gained in laboratories. In labs, the experiment findings are different from those that you can get in natural environment in the exclusion zone, for example. And of late, we've been working with our Norwegian and Ukrainian colleagues in the zone, in the reserve, and we study fish. And if we look at fish in aquariums and it depends on the feed. As much the feed is contaminated, so much the fish would be contaminated. But you cannot duplicate or restore the situation in the natural water bodies in the aquarium. Now, and our research has concluded that with the dose of under 100 micrograde per hour for ground systems and aquatic systems at under 400 micrograde per hour. Certain changes would not occur in the population. So on the majority of the contamination zone, we have lower indicators, lower values. For water bodies and only in the red forest can this dose be higher than 100 micrograde per hour. It's difficult to study the impact of ionizing radiation on the living organisms with such low doses. There are other factors that will tell on those organisms, and it would be next to impossible to say whether it is the impact of radiation or other factors. For example, many study the loss of apical domination in the populations of pine trees. And our colleagues who work there focusing on this very issue, and they now work in Fukushima, and they found out that the situation is very similar. But in Chernobyl zone, it is difficult to see whether those were reindeers or any other deers who picked up or chipped off the tips of such young pine trees. So the exclusion zone, at least I see it as such, is a unique test field for research. And the most interesting research that should and can be continued is the migration and behavior of radionuclides, and then the dose loadings on human bodies, etc. Today, many study radioecological effects, but the dose loading is difficult to assess and to quantify, in particular in the fish. Just they get internal doses, and you will find radionuclides in bones and in flesh, but it's difficult to assess the external impact. Some of our colleagues conclude that corpse, for example, get the major dose during the wintertime, during hibernation, but we don't know how deep under in the sill they are. And the depth is, say, 35 centimeters or more. So the timeline, the depth of the sediment, as they say, then the duration, and in August the concentration of oxygen is so high that, and the ammonium concentration is so high that fish cannot survive there. So we have to study that in real, natural conditions, and we are planning to carry out such research. And one should know that the time when science was advanced in the offices, at the table, and in order to obtain the reliable data you need very cutting edge equipment and material based. So for us to carry out true in-depth research, we need assistance on the part of our foreign colleagues. Okay, we've discussed extensively the nature and character of radioactive contamination in Chernobyl and in Fukushima, and we said that those two are different. Can we say that these two zones complement each other in terms of the radio ecological research? And we know that in Fukushima there is a large vast areas of ocean that is contaminated. Yeah, we can use these two areas as mutually complementary in some places, but not always. For example, when it comes to the dose of radiation or exposures, so it doesn't depend on the form of sedation and fallout. And dose shows the impact on any living organism. As far as the migration and combination of produce, again, we can use findings obtained in Chernobyl and extrapolate them to the results to be obtained in Japan and Fukushima. Thank you. Now, in terms of radioecological research, we all understand that it's important to assess doses and exposures. Where can these findings be applied if we go beyond the zone of radioactive contamination? You know, these findings are essential for the liquidation of or a rescue after the already existing or the aftermath of accidents. For example, if we want to rezone certain settlements in Kiev region, where over the last 20 years the dose does not go over 5 millisievert. And people get that from the natural background contamination. But when we speak with the authorities, they say you do not take on board the impact of plutonium, of americium, etc. No, we do. But in order to substantiate our approach, we have to get these findings and these data, which are the results of radioecological research. Besides, we should be prepared for any emergencies that might occur. We should ensure our preparedness. Unfortunately, prior to Chernobyl accident, we didn't have any idea of how could the radioactive materials behave. We thought that strontium will behave like in Chelyabinsk, with a huge wash out into the waters, and we did a lot to prevent the wash out of radionuclides into the aquatic systems. We built dams, etc. If we knew that strontium was part of the radioactive particles in the fuel, we wouldn't have had to do all of that. It's like water in a bottle. Until you open it, it won't be pouring out. So only after some time did those particles start to decontaminate and go into the surface waters, into the groundwaters, and on the surface plantation. But that took a very long time. And in the sediment and the silt, those particles are conserved, and they do not affect the quality of the water.